Right, hello everyone. Thank you for joining this worldwide lab meeting for the Psychiatric Genomics Consortium. So we're delighted to welcome you here today. I'm Catherine Lewis. I'm an Executive Director of the PGC and I'm Professor of Statistical Genetics at King's College London. So the worldwide lab meetings are the PGC's regular scientific meetings, and they form an online platform to hear about the exciting research that's going on both inside and outside the PGC. And if you've got suggestions of what you would like to hear about or what you'd like to offer to talk on, please do let me know. The sessions are welcome to everyone or open to everyone, so please do forward information to your colleagues and your collaborators. And the recordings are placed on the YouTube channel for the PGC immediately afterwards. So for today's Worldwide Lab meeting, we're delighted to welcome representative of our newest working group in the PGC, that's the Functional Genomics Group, and that's Suyang Yao from the Karolinska Institute. And Suyang's an assistant professor in the Department of Medical Epidemiology and Biostatistics at the Karolinska, and she's interested in integrating genetics with epidemiology and neurobiological data to gain understanding of schizophrenia mechanisms and heterogeneity. And so today she's going to talk on connecting genomic results for psychiatric disorders to human brain cell types and regions, which reveals convergence with functional connectivity. And she's going to talk for about 40 minutes, leaving us with plenty of time for Q&A afterwards. So thank you very much for joining us, Suyang, and I will hand over to you.
Thank you so much, Catherine, for the very kind introduction. I'm going to share my screen first. Can you see it well?
Perfect. Yep, just the slides. All good.
Okay. Hello, everyone, and it's my great pleasure to be here and share with you our recently published work, which is titled Connecting Genomic Results for Psychiatric Disorders to Human Brain Cell Types and Regions Reveals Convergence with Functional Connectivity. And in this talk, I'm going to prioritize schizophrenia findings mainly, so that's why I'm changing the title a little bit. Yeah, and here's the QR code for the paper and link. So this paper was earlier published on Nature Communications. Yeah. Okay, so I'm going to start a little bit from the schizophrenia symptoms. So schizophrenia is marked by positive symptoms, which are symptoms added on to the normal life, such as hallucinations and delusions, and negative symptoms, which are symptoms that are taken away from everyday life, such as flattened emotion and lack of drive, and also cognitive and social impairment. And here I'd like to show some pictures from Louis Wayne, who is an illustrator of cats. And he unfortunately got affected by schizophrenia and has some changing of his style in terms of his drawing. What do we know about schizophrenia? So there are lots of research going on in terms of risk factors, but currently the most consistent results, I would say, come from the genetic research. And thanks to international collaborations such as PGC, that we are able to collect enough or great sample size to study the genetic effect. And in the PGC3 study published several years ago, and there have been over 280 loci identified for schizophrenia. And if we zoom out a little bit, to talk about the genetic architecture of schizophrenia. So twin and family pedigree studies have shown a stable heritability around 60 to 80% across different populations. And as we just saw in the Manhattan plot, schizophrenia is highly polygenic, but they're also affected by larger effects of rare variants. For instance, in the schema study, there are rare variants that identified from whole exome sequencing with large effect size, as well as seen in these studies. And the GWAS results are mostly here. Nevertheless, due to its high frequency, most of the patients are still affected by the common genetic variants identified in GWAS. And this argues for the next step, that is to understand the findings from the GWAS results. So to tackle this challenge, one option is to link the SNPs that are identified from GWAS to genes. And here is an oversimplified illustration. So imagine this is the genome, and this colored segments are the genes that are expressed. So we could just map by location in terms of whether the SNP is located inside a gene or nearby a gene. But then the next question is that gene expression is dependent on which cell type it belongs to. So I took this illustration from online, and I think it's nicely shown that if you just take a small piece of the cerebral cortex and zoom into it, we can see the nice 6-layer structures consists of many different cell types. So if this is the gene expression in one cell type, other cell types could have very different gene expression patterns. So how do we tackle this challenge in terms of linking SNP to genes? And single-cell RNA sequencing can really help us here. So traditionally, RNA sequencing has been done in bulk tissues, which is still very valuable nowadays, especially for tissues with a higher homogeneity. But for tissues like the human brain that consists of very many diverse cell types, single-cell or single nuclear RNA sequencing can give us a better resolution. So by measuring gene expression per cell, we could then, based on the per cell gene expression profile, group the cells into cell types. And this is such an atlas from the mouse and published in 2018. So after we have the cell types, we can then zoom into each cell type and study the gene expression over there. Earlier work have came up with this measurement called gene expression specificity. So basically for each cell type, the gene expression has been normalized to a standard like 1 million transcripts. And then per gene, their expression were then compared across the cell types to see if the gene is particularly specific or highly expressed in a certain cell type. So this formula basically summarized this concept. So by comparing between genes in a cell type and afterwards for the same gene across cell type, we can figure out which genes are the most specific for each cell type. So in that case, we could get a specifically expressed cell gene list for each cell type and then start to map the SNPs into the gene list. And we use the widely used partition LD score regression, which basically, in a very simplified version, estimates the genetic signal or SNP heritability in the selected cell type, in the selected gene list versus the SNP heritability per SNP over the whole genome. So based on this concept, the previous generation of our groups have published nice papers and highlighting cell types related with psychiatric and neurological diseases. But then back then, the limitation would be that these studies were only based on mouse data or mouse brain and central nervous data because human data back then were not available. And luckily, 2 years ago, there was a big study coming out, and it's really for the first time picture the adult human brain cell types. So researchers in this study have gathered 106 sections from 4 donors all over the brain, and with over 3 million cells or nuclei, they have found 461 clusters, which could be like broadly classified into 31 superclusters. So in this panel B, you can see the nice map of these clusters. So the upper part of this are the neuronal superclusters, whereas below the dashed line are the non-neuronal superclusters. So this is the data we're using to map the SNP heritability to the cell types. And in our paper, in addition to schizophrenia, we actually looked at a bunch of traits with sufficiently powered GWAS studies. So in this plot on the x-axis, we have different traits that we gathered and QC'd, including psychiatric traits such as schizophrenia, bipolar, depression, ADHD, etc., and brain traits such as educational attainment, IQ, neuroticism, etc., neurological traits, structural MRI traits, and some control traits related to height, BMI, and some blood traits. And on the y-axis are the 31 superclusters that you just saw on the map. And these are just their brief names, such as upper layer upper layer excitatory neurons and deep layer excitatory neurons, etc. And we also have inhibitory neurons, including the interneurons, etc. And then below the dashed line are the non-neuronal cell types. And if the enrichment is significant after multiple testing correction, then we see a big circle, otherwise it's a small circle. And the color indicates the level of significance. So we saw a lot of signals in the neuronal cell types for the brain traits. Whereas as expected, for instance, for multiple sclerosis, we saw signals in the microglia. And also, we saw like microglia signals for the control traits. So the first column is the enriched cell types or superclusters for schizophrenia. We saw many neuronal cell types for the same trait, but also like for we see similar patterns between different traits. So we'd like to ask further. So why do we see this patterns of neuronal cell types? Is it because that they are very similar? So the next step we took was to look at the similarity of the top specific genes across the neuronal superclusters. So this is a heat map made according to the overlap of the gene leads between different superclusters. So the matrix Jacob index basically measures the proportion of the intersection versus the union. And if it's 1, then it's completely overlap. And if it's 0, then the two sets are completely separate. And the highest value we found is around 0.5, and it's between the upper layer and deeper layer excitatory neurons. And excitatory neurons have higher similarity in general, but still the values are not super high. So the answer to this question is that we saw many cell types, but this is not because they are very similar to each other. And then we decide to further perform conditional analysis to account for the similarity in terms of the signal. So here are the neuronal cell types in the 31 superclusters. And what we did is that for each of the supercluster, we condition for the other for another supercluster and see if the signal goes away. So the diagonal indicates whether this supercluster was significantly enriched of schizophrenia heritability from the beginning. And then, for instance, it's the upper layer excitatory neurons was significant, but when we condition on the deeper layer excitatory neuron, the significance goes away, meaning that these two cell types may have some statistical dependency with each other. But still, for some cell types, no matter what other cell type are included in the conditional model, they are still significant. And those are the deep layer excitatory neurons and excitatory neurons in the hippocampal C1 to 3 regions, amygdala excitatory neurons, and inhibitory neurons, including the CGE interneurons and eccentric MSNs. But here we would also like to highlight that although this is like a conditional analysis, etc., but we are really accounting for the statistical dependence here. Even though like this upper layer excitatory neurons signal goes away when conditioned on another excitatory neuron, doesn't mean that this cell type is not important biologically. So this is the conditional analysis or effort we did at the cell type level. And we also did conditional analysis at the trait level. So here we are trying to answer that if we account for the shared genetic component between schizophrenia and another trait, do we still see similar patterns of cell types enriched? And here, the other traits we accounted for include the bipolar disorder, education attainment, IQ, depression, neuroticism. So this black bar in each of the cell type group or cell types indicate original significance of schizophrenia. And if it's a solid color, then it's significant. Otherwise, it's not significant after multiple testing correction. And here I'd like to draw your attention to two examples. So the first is still this upper layer excitatory neurons. So originally it was significant, and after we account for bipolar genetic signals, the bar is not significant. And this means that the fact that we saw the same cell type for two traits might be due to that these two traits have similar mechanism, or their genetic effect have similar impact on the gene expression of the cell type. Whereas another example is this hippocampal C1 to 3 excitatory neurons. So no matter what other traits we are trying to subscribe the genetic effects from schizophrenia, we still see the significant enrichment, meaning that this cell type is suggested by the genetic liability of these traits due to perhaps different etiologies. Okay, so far are the results at supercluster level. And next, we went deeper into the cluster level. So because we only found signal in the neuronal superclusters, I'm showing you the results of the neuronal clusters at the slide for schizophrenia genetic risk. And so in this, this t-SNE was originally from the Slate et al. 2023 paper. So each dot on this t-SNE map is a cell, and they are clustered together into the superclusters. And here we can appreciate the change within the supercluster in terms of the schizophrenia genetic risk enrichment. So the darker the color is, the more significant is the enrichment. So the strongest signal we see is some neuronal clusters within the amygdala excitatory superclusters. And also the hippocampal C1 to 3 excitatory neurons also in general have a high degrees of significance of enrichment. Yeah. And we also performed a similar analysis as you just saw in the bar plot. So if we subtract the heritability or SNP effect from bipolar and from IQ, what would we see in the same plot? So if we subtract the bipolar disorder genetic effect from schizophrenia, we see that a lot of signals goes away or become weaker, and this is potentially because the shared genetic component between schizophrenia and bipolar are implicating these findings. However, in contrast, if we subtract the genetic effect of IQ from schizophrenia, a lot of the signals remained, meaning that the same cell types got found in both traits, perhaps due to different etiologies. Okay, so far we have been looking at the schizophrenia and other traits heritability enriched at cell type level. But we also want to take advantage of the information on regions and level this up to brain regions. And remember this nice illustration. We also looked into this origin data set in terms of the cell type distribution. So on the x-axis are the 461 cell types or cell clusters that are grouped into the 31 superclusters, also indicated by its color. And on the y-axis are the brain regions. For instance, we have frontal cortex, limbic cortex, etc. And each of the small dots are one of the 106 brain regions that were sampled in the brain atlas data. So here we can see some patterns. Still, the dashed line indicates a separation of neuronal cell types and non-neuronal cell types. So for non-neuronal cell types, they have a pretty even distribution across different brain regions. However, we can really see some patterns in terms of neuronal cell types distribution. For instance, in the cerebral cortex, there are like this pattern of upper and deeper layer excitatory neurons and inhibitory neurons, whereas in hippocampus, we see a different pattern. And in amygdala, they also have specific excitatory neurons for the amygdala structure. Okay, so the next thing we do is that we use the weighted sum approach to leverage the cell type level information to brain regions. And here is a simplified example. Say we have region 1, and it has 4 neuronal clusters, and this is their proportion, and this was their cell type level enrichment of schizophrenia heritability. Then we just weight their enrichment z-score with the proportion and sum up. And this is a z-score at regional level we got. And we then standardized it and calculated the p-value at regional level. And here is the results for schizophrenia at brain region level. So to keep the color scale inconsistent, the reddish color indicates higher level of significance, and the yellowish color indicates lower significance. So we see that pretty much the whole brain are affected by the genetic risk of schizophrenia. And this is what the cerebral cortex looks like. But when you turn it around and look deep, like in the deeper structures, you can see that there's a higher variation. Actually, the highest or the most significant enrichment we found was in the amygdala and the hippocampus. So in this plot, we can see the tails, body, and the head of the hippocampus and the nuclei in the amygdala. And this is the cortical amygdala and the lateral structure of the amygdala. And linking it back to the cell type level, we wanted to ask like where the signals come from. So here, like for the hippocampus, the outer layer indicates the significance level, and the middle layer indicates the supercluster of the cell types, and the most inner part indicates the bigger or broader cluster or classes if it's an excitatory neuron, inhibitory neuron, or mixture. So for the hippocampus, most of the signals come from excitatory neurons, whereas for the amygdala, there's more even distribution of the signals between excitatory and inhibitory neurons. So to this step, we have leveraged the cell type information to a region level, but we want to like further validate this regional level findings. So we ask, what about their functional connectivity? What about the like connections between these brain regions? So the next step we do is that we try to validate it, use a orthogonal approach, that is the fMRI data. So this part of the analysis is independent from the like previous transcriptomic data. So we have two data sets in case control settings with 42 plus 42 case and controls, and another is the COBRA data set with 54 cases plus controls. So what we do to start with is that because we can, in imaging data, we can find different regions of interest in the brain. There are too many, so we want to first limit the number of regions we look at. And here we took all the regions that showed the significance in our prior step. But after that step, we don't pay attention to their specific enrichment score anymore. So from the previous step, there are 38 regions, and in the fMRI data, we have two sides of the brain, so we double the size. So we start with 76 brain regions and the case control setting. And then what we do is that we take the connection matrix of the 76 brain regions. So we first start from a 76 by 76 connection matrix, and then we feed this matrix to a deep neuronal network classifier called BRAF2. So what this classifier does is that based on each person's connectivity matrix, it would try to predict the case and the control status of this person, if it's case or control. And so here we don't care too much about the output, if it's classified very accurately or not. What we really care about is their estimation of how important each of the connection is. So corresponding to the connectivity matrix, we also get a feature importance matrix measuring how much this feature is contributing to the final classification. And by summing up all the feature importance of one region, we could then tell if a region is important in this type of prediction. And then we go back to this list of 76 regions and rank them and see who contributes most, who contribute least. And then we remove the least contributing region and start this loop again. So in this recursive feature elimination procedure, we remove one region each step until we don't have more regions to remove. So the key point of this analysis is twofold. So first is that the important regions tend to be retained to later stages. And also the important functional connectivities will be suggested by the feature importance matrix. And we separated the analysis for these two data sets, and in each data set, we ran a 5-fold cross-validation procedure. And this is the result in terms of regions and functional connectivity. So to the left is the regional level results. We basically mark these regions in terms of if they are amygdala region, if they are hippocampus region, if they are cortical region, or other basal forebrain regions. And in both of the data sets, the amygdala and hippocampal regions tend to stay until the later stages. And to the right side is the feature importance matrix that measures how the functional connectivity is contributing to the classification. So here are the heat map with the darker color indicating that this feature is contributing a lot to the model. So we could see that there are some connections that are contributing a lot, and then we distinguish if this connection is involving any amygdala or hippocampal region. And if it's yes, then it's a red bar. If it's no, it's a blue bar. So in this two data sets, here you see the feature importance value, and they are ranked in descending order. So in both data sets, the feature importance involving amygdala or hippocampal regions are greater than the feature importance that are not involving these two structures. And this indicates the importance of the amygdala and the hippocampal regions that we saw in the transcriptomic data. Yes, to summarize this work, we basically do the exploration in 3 steps. So the first is that we take the very important study of the human brain cell types, as well as the GWAS summary statistics, and try to infer the SNP heritabilities enriched in different cell types. And in the second step, we leverage this based on cell type distributions in the brain to brain regions and pinpoint specific regions that are potentially most disturbed by the genetic risks. And finally, we validated this regional level results using independent fMRI data. And in terms of conclusions, for schizophrenia specifically, that the genetic risk pinpoints to neuronal cell types across many brain regions. And also our results highlight that amygdala and hippocampus is important in schizophrenia mechanism, and maybe future transcriptomic studies can look further into these regions. And finally, we found convergent evidence between genomics and brain imaging. And with that, I'd like to thank my fantastic co-authors. It's been really cool to work with these brilliant people. And also thanks to the GWAS data providers and the RNA sequencing data providers, as well as the fMRI data and donors and participants. And I'd also like to take this chance to do a little ad for myself. So I've recently become independent and focusing on risk factors for schizophrenia and the underlying mechanisms. And if you're interested, please reach out, and I'm very happy to talk about this. Yeah, thank you very much.
Great. Thank you very much, Shiyang. That was fantastic. A great whistle-stop tour of the brain and all the fantastic work you've done, and such great visualizations that really helped people understand what you're doing. Thank you. So we are now open for questions. I've suggested that we use the chat for questions if people are happy with that, because that means everyone can see them. Whereas if we put them in Q&A, then I think only those of us that are panelists can see them. So please do put your questions into the chat if you're happy to do that. So maybe I will. So I'll kick off. Shuyang, while other people are thinking what they want to ask. So you've shown just how much information you can get when you integrate these different sources of information, from from expression and genetics, and then the neuroimaging. What's the current limitation here? What's not available that you really wish was available, either technologically or access to reference data that would allow you to take your work to the next step?
Yeah, thanks for this great question. And in terms of limitation, I think there are several aspects to think about. So one thing is that regarding the brain atlas data, so this is really the first comprehensive data we have, and it's really comprehensive, covering 106 brain regions. But still, there are still regions not being sampled, and the cell types or their expression pattern, we still don't know, and it would be great to have. And also another thing is regarding this method. From what I read and my experience, this type of enrichment analysis is mainly based on comparing between different cell types. So what we include initially in this comparison really matters in terms of how specific or what specificity each gene would get for each cell type. So I think this is a limitation. Yeah, I also talk about this with my colleagues, and discuss about this limitation, and so far we think this relative feature of this measure is being seen in many papers.
Oh, interesting. Right. Thank you very much. And there's a few questions in the chat. So the first one, I think it's from Mark Adams in Edinburgh, and he's asking if you could explain again how to do the cell enrichment analysis that adjusts one phenotype for another. So I think you showed schizophrenia adjusted for bipolar and then IQ, right?
Yeah.
And your slides are still up if you wanted to scroll back and...
Yeah, of course. Sorry, I didn't talk in detail about how we did this analysis. Either the bar or this t-SNP plot will be fine. So what we did was the genomic SEM. We basically used this method to subtract one traits like a SNP or GWAS signal from the other traits, and then use the unique component per SNP to do this estimation.
Okay, right. Hopefully that's okay for you, Mark, but if you want to come back and add a further question, please do. The next question is from Jens, who's asking, what's the next step and what would be confirmation of these findings?
Yeah, that's a big question. I think there are many next steps to be taken. So first, for instance, that we, from transcriptomic data integrated with genetics, we could highlight some brain structures like the amygdala and the hippocampus. I think one of the next steps would be like dig deeper into the transcriptomic of these regions to see what cell types are there and what gene expression are there. Actually, last week, I believe, there is a new paper on Nature from Jian Yang's group, and they were doing this spatial mapping and using their new technology. I think it's very cool. They also were pointing, for instance, to glutamatergic neurons in the dorsal hippocampal region. So it's sort of pointing to similar targets as our findings. And I'm personally really interested in the connectome as well. So it would be really cool to be able to tell which cells are connected to each other based on their expression profile. And then we could see, for instance, if there is a path that we could map to, and if this path is enriched of the genetic risk, etc. And also, I don't know if I'm expanding too much. I'm
No, go ahead.
thinking in terms of timeline, like this developmental stages in different ages, for instance, could also be interesting because some of the function impairment during adult we see may actually be a result of developmental disturbance during, for instance, adolescence, childhood, and I think there's a lot to be explored over there.
So you'd want to be able to pinpoint these to particular developmental stages, is that?
Yeah, that would be great, which we can't do with cross-sectional data at adult stage.
And so what would you need to be able to do that?
I think that would be like a transcriptomic atlas during different brains, during different ages and life stages.
Okay, right, thank you. And then the next question from Cynthia is asking if you're considering epigenomics to find regulatory elements that are associated with schizophrenia.
Yeah, I think, this is also a great question. And yes, we are actually. So Arvid, our co-author, has recently run some analysis using the open chromatin data, ATAC-seq data, I believe was also published in the same year as this brain atlas. And they have single cell ATAC-seq data from different brain samples covering several different brain regions. And there, the strongest signal was also in the excitatory neurons. Yeah, and in neuronal cell types.
Interesting, right? Thank you. And then Sylvanus complimented you on a great talk, which we all agree with, and was asking whether you'd expect similar magnitude of cell type enrichment as at the SNP versus the gene level.
Thanks for the comment and question. And I'm not sure I have a clear understanding about the similar magnitude of enrichment. Yeah, or maybe this question. Sorry.
No, go on.
I have a quick thought about maybe this question can be thought of from the perspective of SNP location inside or outside of the gene. I don't know if please correct me.
Do come back and add more, Sylvanus, if we've not answered your question.
Yeah.
Fully.
Yeah, and this reminds me of another limitation I wanted to talk about when you were asking about the limitations. Another thing is that we were mapping SNPs to genes directly by location. So we draw a window around the gene and say, okay, SNPs within this window would be counted. But actually related to this comment, there are more sophisticated ways to do it. If you know the QTL, for instance, if the SNP is linked to certain genes expression, you could include the specific SNPs. But another thing related to that is that eQTL can also have a cell type specificity that currently we don't have a good coverage across the brain. We might have a cell type specific eQTLs for certain cell types, but in this particular study, it's not available for the cell types.
Right. Thank you. And I think Sylvanus has added, he's thinking more of SNP heritability versus magma enrichment analysis here, but maybe you've now filled in that in your last comments.
Yeah, we also tested, actually our co-author from University of Queensland in this paper have done a thorough comparison between different types of this type of enrichment analysis, including our top specific genes plus stratified LTC versus magma, etc. And I think, as we said, this type of analysis is really sensitive to what data you start with. For instance, this is a relative measure, etc. So we don't know the ground truth here. So in my opinion, it's like a hard comparison.
Right, a good point. Thank you. So do keep adding the questions if other people have things that they want Shuyang's expertise on. But maybe I can pick up on, so at the very end, you highlight that we're looking at neuronal cell types here, but they're across many brain regions. So, that's really interesting. There's lots of spread effects, but where does that tell us to focus next, really? Because having this across lots of brain regions isn't terribly helpful, is it? If we, really want to be able to focus on mechanism a little more explicitly.
Yeah, exactly. I also have the same question in terms of, it's so many cell types are popping up, influenced by the genetic risk, etc. Yeah, so as we just discussed, one thing that could be focused on with clearer indications, probably the amygdala and hippocampal regions. Yeah, but for the others, I don't have a good answer, I think.
Okay, fair point. And then I think Andrew McIntosh has asked, could you look at the intersection across all psychiatric disorders?
Yeah, that's a very interesting suggestion, opinion as well. Do you mean like something like the general factor, for instance? Yeah.
So I suppose you could use the P factor or the new work coming out of the a cross disorder group, for example. Is that,
Yeah,
was that what
definitely.
you meant? Or Andrew's suggesting like schizophrenia, MDD and bipolar disorder, so more of a focused subset.
Yeah, definitely. I think that's a great suggestion. Good idea. And I believe in the PGC bipolar work recently, something related to this has been done, like comparing schizophrenia and bipolar GWAS signal. Yeah, in terms of that, of the cell type enrichment.
Yeah. And you've talked through this very nicely for schizophrenia. If other people on this call are inspired and want to apply these methods and these tools to their particular disorder or trait, what's the first steps? Where can they go to for help there?
Yeah, actually, we have done the analysis. It's just we haven't visualized it for other regions. Sorry, in terms of brain regional level results. So we have the results in the supplementary tables of this paper. Actually, in terms of visualization, I've been using the tool that is recommended by the Allen Brain Institute in terms of visualizing their 3D model. And you basically plug in a label, then you could like see these videos in the, see this 3D models in different colors for some other traits, for instance.
Okay, great. Thank you. And I've not looked at the paper in detail, I admit, but you've chosen to focus on schizophrenia, which obviously, you know, we have it has a high heritability. We have a lot of GWAS findings for. Could you give us a sort of picture of how that differs when you start to look at something more challenging, more polymorphic, with lower heritability like MDD?
Yeah, that's also a good point. Maybe I can come back to the first.
Yeah.
Sure. Yeah, we actually have a selection criteria for the traits, like several criteria to ensure that they have sufficiently powered GWAS, like they have to have at least 10 significant loci, for instance. And we also plot the sleep heritability on top. And actually, we also run a analysis that we didn't show here, is that we plot the number of cell types that we found in relation to the year of the GWAS publication, because if it's published later, and then they usually have greater power. You know, we have a plot regarding that, so it's also analysis relevant to the statistical power in the GWAS.
Oh, and I think we've used similar things in our MDD paper, haven't we, showing how it strengthens over
Right.
time, which is really helpful as support. Thank you. That's really interesting. And so, yeah, it's interesting. The ones that it's interesting that MDD does come up with a similar number of regions, doesn't it, despite the lower heritability? Thank you. That's reassuring. So I've put in the chat a last call for questions. Is there any final question for Shuyang? Or is there anything you wanted to add, Shuyang, as we've been discussing it? Is there any final points you want to make about this really exciting area and your excellent work?
Yeah, thanks. I feel very lucky to be able to work on this project. It's also a great experience for me. Oh, yeah.
Right. Oh, and one final question then, I think from Sylvanus again, asking about how the findings would help with drug development.
Yeah, that's also a nice question. Can be related to next step questions. Yeah, I think maybe also related to your question regarding so many cell types, etc., that's got highlighted. Maybe we can look for some overlap regarding like existing drug targets and the specifically expressed genes per cell type. Maybe the overlap may give some interesting insights.
And then Cynthia's asking if you could please share the link to your paper in the chat for those of us that missed the.
Sure, I'm going to do that now. You see it?
Yeah. Yes. Okay. Super. Yep, and that works perfectly. Lovely. Then we will finish there. Thank you very much for a fantastic talk. I think you could see by the number of questions and the interest in all of this work, just how excited everyone is by what you're starting to find in this area. I'm wishing you the best of luck as you establish your independent group and make many further strides here, I'm sure. So as always, the recording will be on the YouTube PGC channel shortly, and thank you everyone for joining. We'll finish there. Goodbye.
Thank you. Bye.